Coming to the next lecture of Journal Embryology, we will discuss these competencies, that is, describe the stages and consequences of fertilization, describe cleavage and formation of blastocyst, describe the process of implantation and common abnormal sites of implantation. Now, fertilization is the process of fusion of male and female gametes, which are basically mature germ cells with haploid number of chromosomes. That's why we call them as haploid cells, to form a single cell called as zygote. Now, this fertilization is occurring within the 24 hours of ovulation in the ampulla portion of the fallopian tubes, which is the most dilated portion. Now, the results of fertilization is it is going to determine the genetic sex of the embryo, restoration of the diploid number of chromosomes, initiation of the cleavage. It happens, then it is necessary for the propagation of the species and their evolution. Now, these are the steps of fertilization. What are the different steps which are happening? First is the transport of gametes into the uterine tube. Then fusion of the gamete, it happens. That is, you can see here, then the result of the fertilization is there. What is that? That is in the form of zygote. So these are the things which are happening in the fertilization. Now, first is the transport of gametes into the uterine tube. So only 1% of sperms deposited in vagina enter the cervix, where they survive for many hours. They travel from vagina to ampulla of fallopian tubes for fertilization. You can see in this diagram. So they are just traveling from the uterine cavity and towards the fallopian tube. Then the movement of the sperm from the cervix to the uterine tube, it occurs because of the muscular contractions of the uterus. Then muscular contraction of the uterine tubes, it basically they help in the movement of the sperm towards the ampulla portion of the fallopian tube. Now, oocyte reach the tube by contraction of the uterine tube and ciliary action of the epithelium. Now, the prostaglandins present in the semen causes contraction of the uterine muscles along with the oxytocins, which is released by the pituitary, promote the journey of sperm. Now, next step is fusion of gametes. So, out of 300 to 400 sperms reaching the uterine tube, only one of them fertilizes the egg. So, what are the barriers which are crossed by sperm before fusion? That is, you can see in this diagram, the corona radiata, then zona pellucida, then vitelline membrane. So, these are the three membranes which are basically crossed by the sperm for fertilization. Then sperms get ability to pass through these barriers by two processes, that is capacitation and acrosomal reaction. So what is capacitation? It is the process of conditioning of sperm in the female genital tract, which lasts for about 7 hours. In this process, the glycoprotein coat of the plasma membrane overlying the acrosomal region of the spermatozoa is removed. Only these capacitated spermatozoa, they can undergo acrosome reaction. It is a slow process which enhances the sperm motility and receptivity. You can see here. So these are the things which are happening in capacitation. Basically, this is the conditioning of the sperm. Now, acrosome reaction is, it occurs when capacitated sperm come in contact with the zona pellucida. So it is induced by the zona proteins, which results in the release of the enzymes, that is hyaluronidase and acrosin, from acrosomal cap. So this is the acrosomal cap from where these proteolytic enzymes, they are released. So it is a rapid process with outcome of penetration of the zona pellucida. Now, chemical which are disintegrating the barrier, or which are basically helping in crossing the barriers, are hyaluronidase. It helps the sperm to cross the corona radiata. Then acrosin is going to help basically to penetrate the zona pellucida. Integrin peptides, they just help in penetrating the vitelline membrane. Now, the results of fertilization is, if we see the process, what are the three steps? We have discussed that. Now, what is the result of fertilization? Third step is results of fertilization. The female gamete completes second meiotic division, that we know. As a result, a mature ovum is formed with the second polar body in the perivitelline space. This is a perivitelline space. Then his first polar body also divides into two polar bodies. So total 3 polar bodies are seen in perivitelline space. So these are the things which are going to happen. Then formation of male and female pronucleus. You can see in this diagram. Then diploid number of the chromosome is restored in zygote then. So the male and female pronuclei meet and nuclear membranes disappear, resulting in a zygote with diploid number of chromosome. Chromosomes are equally shared by both the parents, but cytoplasm is exclusively from the mother, which is essential for the development. Now, next step is result is chromosomal sex of the offspring is decided by the father. X-bearing spermatozoon with mature ovum forms female child, and Y-bearing spermatozoon with mature ovum forms male child. Now, the beginning of cleavage, before this mitotic division occurs, the chromosomes are arranged at the equator. Now, chromosomes arranged in equator migrate to the opposite pole with equal proportion of the cytoplasm. Now, next step is after this, once the chromosomes are arranged in the equator and they are moving towards their pole, the next step happening is that is two cells basically they are formed and the cleavage is there. Now, this is the process of repeated mitotic segmentation of the zygote within the zona pellucida to give rise to a small cells called blastomeres. It lasts for 6 to 7 days after fertilization, aims at the formation of morula and later on blastocyst. Now you can see the human embryo development. Fertilized egg, 2 cell stage is there, then 4 cell stage, 8 cell stage, and then 16 cell stage, which is given the name morula. Now after that the blastocyst is formed. So this is the sequence, or you can say the sequences of the events, that is zygote, 2 cell stage, 4 cell stage, 8 cell stage, 16 cell stage, and blastocyst. So approximately 30 hours after fertilization, 2 cell stage is achieved. Then approximately 40 hours after fertilization, 4 cell stage is achieved. Then approximately 3 days after fertilization, 8 cell stage is there. Then around 4 days after fertilization, morula is formed, which is going to form the blastocyst. Now in the blastocyst, inner cell of the morula forms the inner cell mass, which forms embryo proper, and outer layer of the cell is called as trophoblast, which forms the placenta. Now next is blastocyst formation. Cells of morula, you can see in the diagram, continuously divide and form the 32 to 64 cell stage. Then uterine fluid slowly enters or diffuses through zona pellucida and gets accumulated in the intercellular space of the inner cell mass. Then accumulated fluid, it forms the cavities that fuse to form a single cavity called as a blastocele or blastocyst cavity. You can see here. Now, outer flattened cells of the blastocele. Now, these are the cells which are going to form the trophoblast, and inner cell mass, it forms the embryoblast. Now, embryoblast, trophoblast, and blastocyst, this blastocyst cavity or blastocele, they form the blastocyst. So, embryoblast, trophoblast, and blastocele or blastocyst cavity, they are going to form the complete blastocyst. Now, the side of the embryoblast attachment is called as embryonic pole, and opposite side is called as abembryonic pole. Now, what is the fate of blastocyst? Embryoblast, they form the embryo. Trophoblast, they helps in the formation of the placenta, and blastocele, it forms the yolk sac. Now, role of zona pellucida is it prevents the embryo from the implantation at abnormal sites, that is in the fallopian tube or uppermost part of the uterine cavity. So, what are the glycoproteins which play an important role is that is ZP1, 2, 3, and 4. Now, how they help in? That is, they help in sperm binding, gamete recognition, penetration, prevention of polyspermy, then induce the acrosomal reaction, then maintain the integrity of the early embryo. Coming to the implantation, this is the process of attachment of blastocyst to the uterine endometrium, and it is the subsequent embedding into the uterine lining in normal event. So it starts in the first week and continues till mid of the second week of development. Now the stages preceding the implantation is that is blastocyst is formed, thinning of zona pellucida it happens, then disappearance of the zona pellucida is there. Then about 6 days after fertilization, the blastocyst attaches to the uterine lining. We call it as endometrium at the embryonic pole. You can see here, this is embryonic pole, this is abembryonic pole. So the stages of implantation is, first is decidual reaction it happen, we will discuss in next lecture in detail. Then hatching of the blastocyst it happens. Then adhesion of the polar trophoblast, this is the polar trophoblast to the columnar epithelium. Then penetration of the blastocyst through the uterine epithelium. Then erosion of the endometrium. The proteolytic enzymes from the trophoblast and uterine epithelium erode the endometrium and embedding in the blastocyst deeper and deeper into the uterine mucosa to lie within the thickness of endometrium. Now the trophoblast differentiate into, you can see here trophoblast is differentiated into cytotrophoblast and syncytotrophoblast. So the part which is embedding within the endometrium, so that is undergoing these changes. Now next is closure of the penetration defect in the uterine epithelium by ninth day. So this epithelium is reformed. So whole of the blastocyst is basically is present within the wall of the uterus. So the completion of the embedding, it happens by 12th week. Blastocyst is fully embedded and establishes the nutritive relationship with the maternal blood vessels. So these are the steps of implantation. Now, what are the types of implantation? Central implantation, blastocyst is implanted in uterine cavity, so it is seen in carnivores. Then eccentric implantation is blastocyst is implanted in uterine crypt, that is seen in mouse. And in human, the interstitial implantation is there. Blastocyst is implanted in the uterine endometrium. Now, clinical correlation is the normal site of implantation is upper part of the body of the uterus in mid sagittal plane in the posterior wall in 50% cases, and in 45% it is on the anterior wall. But abnormal site of implantation may be there. If it is in the lower uterine segment we call it as a placenta previa. Then extrauterine site we call it as ectopic pregnancy. In extrauterine site, these are maybe in the tube, they may be in the abdomen, or they may be in the ovary. So in tubal implantation, the most common is interstitial, and second is ampulla, and third is isthmic. In abdominal, it is of two type, primary and secondary. And ovarian implantation, it is rare and can cause teratoma. So what are the complications due to implantation at ectopic site is tubal or uterine rupture, massive hemorrhage, shock, then DIC may happen, that is disseminated intravascular coagulopathy, then death may occur. So these are the complications due to implantation at ectopic sites. Now, what are the complications due to surgery for ectopic pregnancy? If the surgery is done for ectopic pregnancy, the excessive bleeding may happen, infection may be there, injury to bowel, bladder, ureter, major vessel may happen, then leading on to the infertility. So this is all about today's lecture. Thank you very much.